All right. So today what we'll be looking at is protein synthesis and going through it. So if you recall, whenever we talked about cells, we talked about the cells that have specific functions and what those functions were. Um, the ribosome, its primary function was protein synthesis. That's its job, is to make proteins. So that's what we're talking about during this. Is we're going to talk about how, and if you also recall, whenever we talked about what a protein is basically the structural component of the cell that's building all your traits and all of that. So basically, we need to figure out how does the ribosomes know what to make, what kind of proteins to make on what they're building. Much like an architect has to design plans and builders need to have those plans in order to build a house. So that's kind of the mindset to use, and I'll use that analogy a good bit. So please don't forget that all the information always starts with DNA. So we have to figure out how do we get the information that's inside the nucleus to ribosomes so they know what to build. And so that's what we'll be going through today. So first thing we need to know is the relationships that exist between DNA and chromosomes. Please don't forget that DNA is this long double-stranded helix that is bundled up and coiled up together inside of a chromosome. But whenever we take an individual segment of DNA, it codes for a specific gene. It codes for a specific trait that we're seeing, or whether it's blue eyes or whatever that it might be. And so that's what those stripes are on this chromosome in this image. That's what those stripes are. Those would be those alleles. That'd be those genes that are showing up. That's that individual area. And then, of course, the chromosomes, and you have 46 of these chromosomes. So whenever we talk about this process of taking the information from DNA and making proteins using this information, that's this really big term that's called the Central Dogma of molecular biology. Basically, how do the cells make proteins? Because whenever they're making proteins, this is how they are able to maintain their homeostasis. This is how they're able to regulate your body processes and build the traits that are needed, that have been passed down, and how all this occurs. So this is a process. It's a three-step process, and we'll be going through those 3 steps today. So basically, the Central Dogma describes a process by which DNA is replicated. That's the first step. mRNA is transcribed, second step, and tRNA is translated in order to link amino acids together to make proteins. And remember, those proteins are what's making and building your traits, as well as other processes they undergo. So what you see over here, so typically we always, you already know what RNA is, but there's actually different types of RNA. There's mRNA, there's tRNA. There's another one that we really don't talk about very much at this level, and it's called rRNA. And these, of course, are the 3 steps. This is the central dogma, and you will need to know that central dogma very specifically. So the first step is whenever DNA is replicated. So we've actually already talked about this, but now we're actually going to replicate DNA. So please don't forget, DNA, it's only found in the nucleus, so that's of course where replication is going to occur. It happens during the S stage of interphase, if you remember that from whenever we talked about mitosis and the cell cycle. It's considered to be semi-conservative because basically what it is you have that original template of the original strand, and then we build a new strand off of the original. So that's why we call it semi-conservative. And then the enzymes helicase and DNA polymerase. We talked about those in mitosis as well. Remember, helicase is the enzyme that untwisted and unzipped the DNA strand, and then DNA polymerase is the one that added in the new nucleotides. Whenever we talk about those nucleotides and adding them in, this is where we'll be talking about this time, is talking about those base pairs. Remember, adenine will pair with thymine, and cytosine will pair with guanine. And I have an example of to get you to do on the next slide. A new thing that you'll see this term that deals with percent composition. So that just basically means if you had a DNA strand And if 20% of them are adenine, I might ask you what percent of them are thymine. So you might easily want to think real quick on that and say, oh, well, it's 80% because 20, 100 minus 20 would be 80. Remember, since A always pairs with T, that means that what it should be, if there's 20% adenine, then there should be 20% thymine. Just like C pairs with G, well, 20 of 20 is 40, so that leaves us 60 left. So I know that those two have to be 30 and 30 because all four of these should add up to equal 100. And that's how it should go. You will see questions that deal with this type of information as well on your quiz and on your test. So right here is an example of replicating DNA, and typically how I word that is I'll give you a DNA strand and I'll ask you to replicate the DNA strand or give me the complementary DNA strand. So I give you a few here to start you off. So A pairs with T. The T, of course, pairs with A, G with C, and see if you can finish this out. So what would pair with G here? That would be C again. This T, that would be A. This A would be a T. This G would be a C. This C would be a G. T and A T, A T, C a G. So that's basically how that would go, and you would get your strand and you would have to do that. Now you need to make sure that you're paying attention to the question, because I might use other terms, other processes like transcription and translation. Well, you have to actually do that completely differently if I give you something else. So you're going to have to really make sure that you understand what I'm asking you to do. Okay, so do make sure you read those questions carefully on if I'm asking for replication or transcription or translation. So the second step is transcription. So now that we've replicated our DNA strand, what we're going to do now is we're actually going to transcribe it. Transcribe it is whenever we take the DNA strand and now we're going to make the RNA strand. Now the reason why that M is there is we call that messenger RNA. Messenger. The reason why it's called messenger is that this is copying the message directly from the DNA strand. It's going to be copied and then or transcribed, and then from there it's going to be able to leave the nucleus, because remember, DNA can't leave the nucleus. This process does occur in the nucleus. mRNA is also called the codon. mRNA is also called the codon. If you think about scription, to get a script, that's a writing. So if it's a codon, basically it's the code, a writing of that information. It's the coded information. And you also are going to make sure that you use the mRNA to read the codon chart. We'll be talking about that next class. We'll be talking about the codon chart next class. But for right now, we want to make sure that we understand what the three processes are. So if I gave you something like this right here, and this is, it is from the same worksheet. And I would ask you to, on this one right here, of course, you would replicate the DNA strand. But now, since we've already done that, and you can look back at your answers from there and just fill that in. But let's look at this mRNA strand, the codon. If you notice, now we're going to pair A right here with T. A pairs with T. But here's another A. But what about RNA? Does RNA have a T? It does not have a T, so we would put a U there instead, because remember, RNA has a U instead of a T. C would pair with G, and so if this is a G here, you would have a C here, and then you would have a G over here in the codon column. Where this T is, you would go T, A, U. Where this A is, you would go A, T, and then what pairs with T would be an A. A common mistake that students will make here, kind of like up here at the top, they'll think that the U should go with that T, and it never goes that way. A will pair with a T, and a U will pair with an A. U's and T's never pair together, ever. Right here where this G is, that would be G, C, G. Where this C is would be C, G, C. Where this T is, it would be T, A, U. Where this A is, it would be A, T, A. Same thing here is ATA, and then the last one would be CGC. And the last step is translation. Translation is where we're going to take that mRNA strand, and that mRNA strand has left the nucleus and it has gone to the ribosome. So that's what this image shows us here, is we have this ribosome. And really, if you look at this structure where it says tRNA, if you look at this structure, it looks like a funny looking T. So that might help you in remembering that this is the tRNA. Where does this occur? This occurs in the ribosomes. The tRNA is called the anticodon. Remember, the mRNA is called the codon, but the tRNA is called the anticodon. The function of tRNA carries the amino acid. That's what these little dots are up here at the top. It carries these amino acids. And remember, as we connect amino acids together, that's what creates a protein, because a protein is made up of many amino acid chains. Or you could call it a polypeptide. The codons and anticodons are put into sets of 3. If you look at this one, it says UCA. And if you look right here, that U is paired with an A. This C is paired with a G. That A is paired with a U. If we go on down, G, C, C, G, A, U, U, A, G, C, C, G. And that's why they're put together this way. And this will only code for a specific amino acid. And as we get these amino acids together, and they're all these different letters, because remember, we can put them in all different types of combinations, and they make different words or they make different proteins. And that's why we have thousands and thousands of millions of proteins, but we only have 20 amino acids. It's all about changing the combination. So here's an example of this. Now, we've already done the complementary strand and the codon strand. And so really all you're going to do is to do the same thing for the tRNA. What pairs with A, U. What pairs with U, A. What pairs with G, a C. And you just go on through the rest of it. All right? And I'll go ahead and get you the next 3 just to help out there, and you can check your answers. So this would be G, C, G, C. Where this T is, it would be T, A, U, A. A, T, A, U. G, C, G, C. C, G, C, G. T, A, U, A. A, T, U, A, T, A, U, A, T, A, U, C, G, C, G. All right, and we'll be doing some work with this in class as well. And so here's a great image kind of looking at the whole process. Right here, we have DNA that was replicated, and then the next step of this DNA gets transcribed. That would be step 2. And then from there, that mRNA sequence, it leaves the nucleus, and it comes all the way down here to the ribosome, where then it can be translated. So translation, of course, happens down here. And I actually pulled this image from, I'm assuming it was from a test. I could have gotten it off of Google, but I don't think I did. And right here, you see this chain here. This is that chain of amino acids that's making a protein. Here's that tRNA that's bringing that new amino acid down here to link up with its complementary pair. So that's it for day 1. Make sure that you're able to know what the Central Dogma molecular biology is, and also go over each one of those steps about what's happening during replication, transcription, and translation. Make sure you know the role of mRNA and tRNA, specifically their role. What does the M stand for? What does the T stand for? And by the role, let's talk about what its job is to do. Where does protein synthesis happen at? That's just, a general question talking about the ribosomes. Why is DNA even involved? It's the one that has all that information. And don't forget, you'll need to make sure you know what the codon and anticodon is. That'll be very important next class. So next class, what we're going to be looking at is how to read a codon chart and mutations, genetic mutations to be more specific.